Hello, Bio 104 students. In this lecture, we'll be covering the diversity and evolution of plants and fungi. As always, we have an outline for the lecture, and we'll recapitulate these topics at the end of the lecture. All right, in the last lecture, we were talking about the early evolution of life, and I was showing you this phylogeny here of eukaryotes, pointing out that the lineages with the red arrow are the unicellular lineages, and the multicellular lineages essentially are the plants, the fungi, and the animals. And today we're going to be talking about the plants and the fungi, but noting that fungi are more closely related to animals than either are to plants. Okay, now plants, green plants are a monophyletic group. However, within green plants, the green algae, as shown here, are paraphyletic. And land plants that we'll be talking about in much more detail are derived from green algae, in that land plants make green algae paraphyletic. And in the course of the topics we've been talking about in the previous couple of lectures is that the common ancestor of plants are characterized by chloroplasts that resulted from primary endosymbiosis. And then subsequent secondary endosymbiosis happened in two different plant lineages. That is the common ancestor of all green plants, as well as the common ancestor of red algae. But before we dive into our overview of land plants, I'd like to just talk a bit about some of these other groups. So first of all, the glaucophytes, which are the sister lineage to all other green plants, interestingly, their chloroplasts have a peptidoglycan layer, just like we see in cyanobacteria. So this early diverging lineage of green plants, their chloroplasts are more like cyanobacteria than other plant lineage chloroplasts. And then the red algae, there's about 6,000 species of red algae. They're mostly found in tropical ocean habitats. Most are multicellular filamentous organisms. They have complex life cycles, and they only contain chlorophyll A, hence their red color. And if we go looking at the phylogeny of plants again, we're going to be focusing on the land plants here. So the first green plants are what we're calling green algae, and a sample of the green algal diversity is shown here. What is really important here is to point out that green plant evolution, origin and diversification, started in aquatic habitats. They were initially unicellular with flagellae. However, that the early evolution, it's clear the early evolution of green plants is tied to aquatic habitats. Now, terrestrial habitats started turning green about 450 million years ago. So we're looking at here is a geologic time scale, and highlighting here the periods where terrestrialization of different plant and fungal lineages happened. And we see at a time period at about 450 million years ago, we're starting to see the terrestrialization of plants and the ultimate evolution and diversification of the land plants. And in this lecture, we're going to talk about the evolutionary and physiological challenges faced by green plants in the course of their becoming terrestrial. Now, the first land plants likely looked a lot like what we would call bryophytes. Bryophytes are a paraphyletic group that include mosses, liverworts, and hornworts. And they're paraphyletic in that the hornworts are more closely related to vascular plants. So what we're showing you here is the phylogeny of the green plants. We're focusing on the land plant lineage. And let's go ahead and blow this clade up, the land plants. And here we have the land plant phylogeny. So we're showing you here the paraphyletic bryophytes. You have the liverworts, mosses, and hornworts are sister to the vascular plants, all right? Now, what we're going to be talking about with the challenges faced by plants in moving to terrestrial habitats, we're going to think about four things. We're going to think about the challenge of desiccation, that is drying out. We're going to think about the challenge of getting larger. So it benefits plants to get larger. To one hand, we're going to learn to disperse spores, but also to capture more sunlight, in competition sunlight with other individuals around. So plants obviously are not motile, that they, particularly land plants, are not mobile, so they have to, they're sessile. So by getting larger is a response to the competition of competing, or competition for sunlight with individuals around them. And the other challenge is to move water and nutrients throughout the body of the plant. Then the fourth challenge that we'll talk about is, once getting larger, how plants are diversifying structures to intercept more sunlight. Okay, so the first problem of desiccation on land, a very straightforward solution is a cuticle, in that you cover your body with a waxy substance. And essentially, if we look at a cross-section of a leaf, we can see that the cuticle is a waxy layer that prevents water loss from stems and leaves. And all land plants have a cuticle, including bryophytes. Now, but this creates a problem for gas exchange. So if you cover your body with a waxy substance, it's hard then to allow diffusion of CO2 in the body of the plant. So here is an invention that's happened twice in land plant evolution. That is the evolution of stoma, or stomates, where essentially pores, typically in the leaves of the stems, that allow these pores to open and close for gas exchange. Now, the downside of this is loss of water, which having the cuticle was an effort to prevent desiccation. So now you're having the downside of the loss of water. However, this is responded to in plant evolution by the ability for the stoma to regulate, to open and close, and regulating the gas exchange with regard to limiting the amount of water loss. And basically, these stoma are able to work through the osmosis of pumping in or pumping out different ions into these guard cells. So if we have more solutes in the guard cells, then just through osmosis, they'll gain water and they'll close up. And then by pumping them out, we can basically through osmosis get rid of the water, or have the water removed from the guard cells to where the stoma are opening up. So the stoma are allowing for both the gas exchange as well as the opening closing of the stoma is there to limit water loss during gas exchange. Okay, now let's think about the challenge of getting larger. So one way of getting larger, particularly for the non-vascular plants, is to have an upright, what's going to be called the sporophyte. Now, this is the diploid phase of bryophytes, and it's unbranched, okay? Now, what we're going to be learning is that bryophytes have a very interesting difference with regard to the part of the structure that is haploid versus diploid. But in the bryophytes, the sporophyte is the diploid phase of the individual, and this is getting larger as an upright structure that is to limit competition of light with individuals around it. Okay, so this is the problem of how to grow larger. That is to be able to get more sunlight. And how we see this first approached in the evolution of plants is to have an upright branching sporophyte. And these upright branching sporophytes are present in the fossil record dating to about at least 430 million years ago. Now, the third problem involves moving water and nutrients through the plant. And so the solution here, so the bryophytes, the non-vascular plants, are really limited in their size. So they can only grow so large because they don't have a vascular system that allows through the transportation of water and nutrients through the plant, okay? And the solution to this is a vascular system with tracheids that allow water flow through the plant. And the water flow is essentially enhanced through capillary action, really small tubes in the vascular system of the plant. Now, the fourth problem is once we have plants that are growing upright, how to intercept more sunlight. So just like we saw with the stoma, where there's multiple evolutions of stoma in land plants, in a sense, there are multiple evolution of leaves in land plants. And leaves are ways that basically that plants are able to capture more sunlight. So in club mosses and relatives, in a group of non-vascular plant, we have what are the microphylls. So essentially you have an outgrowing of this vascular tissue that becomes this sporangium and a microphyll, and it looks like these leaf-like structures in the club moss. However, all other vascular plants are basically megaphylls. And you have a branching system that becomes progressively reduced and flattened. And you have flat plates of photosynthetic tissue that are developing between the branches. So we're filling in the branches as opposed to filling out from the branches. And the end of these branches evolve to be the veins of leaves of trees, okay? Now, if we look at the phylogeny of land plants, what we see is that the different strategies map onto the phylogenies in this way. So we have the evolution of stoma, we have the evolution of tracheids and a branching sporophyte. And then here we have two different origins of leaves. That is in the early diverging vascular plant club mosses and their relatives, as well as all other vascular plants. So these were the evolutionary solutions that were provided to the challenges that were faced by plants as they were transitioning to a terrestrial lineage. Okay, now with the evolution of vascular plants, there's also a major switch in the life cycle. And I intimated this just a moment ago. So the bryophytes, remember this paraphyletic group, in that the gametophyte, that is the part of the plant that we think of as the body of the plant, is haploid. And this is the dominant phase of the life cycle, meaning that this is the phase of the life cycle that the individual is spending most of the time. And the sporophyte, which grows out of the gametophyte, is actually diploid. And this haploid gametophyte is nourishing this sporophyte. So the sporophyte is the diploid stage. That is, the spores are going to be diploid. Then as we have the evolution of vascular plants, we have a switch in the life cycle where the sporophyte, the diploid phase, is the dominant phase of the organism. And the gametophyte is the haploid phase that is basically a very small part of the life cycle. So sporophytes are large. These are the larger part of the plants. They're gathering sunlight, making sugars through photosynthesis. And the small gametophytes are, they're small, and the sporophyte and gametophyte essentially are nutritionally independent of each other. But the dominant part of the life cycle of the individual becomes diploid, whereas in bryophytes, the dominant phase of the life cycle is haploid. So we could call this the rise of the sporophyte and the fall of the gametophyte. Now let's take a look at the bryophyte life cycle. And what I'm trying to convey to you are the ways that different eukaryotic, multicellular eukaryotic lineages, what they're doing with the diploid and haploid phases of their life cycles, okay? So the Bryophyte life cycle has a number of multicellular, has a diploid multicellular stage and a multicellular haploid stage. So gametes are produced by mitosis, not meiosis. Meiosis produces spores that develop into multicellular haploid organisms. And in bryophytes, fertilization is dependent on water. So what we're seeing here is we have the gametophyte, we have sexual reproduction, and we're going from haploid in blue to diploid. So once fertilization happens, now it is a diploid. And you then have the sporophyte, which is diploid here. And once the spores are released, or I'm sorry, in the sporangium, those diploid spores undergo meiosis. They become haploid. They are released as these ungerminated spores. A spore germinates, it begins to grow, and it's growing to a structure of which the individual will spend most of the life cycle as a gametophyte, all right? And that completes the life cycle. And you can see the life cycle here, the different stages of the life cycle that are diploid in orange versus haploid in blue. So bryophytes are unusual relative to what we think of as organisms. Our own individual cells as diploids, where we spend our lives as diploids, and our sex cells are haploids, right? And the two sex cells come together. In the case of animals, they make a zygote, and that zygote is diploid. So the part of the life cycle in our typical thinking that's haploid is actually very short. It's just the sex cells. But here, the majority of the organism's life cycle is at a haploid stage. It's only diploid for a short period of time after sexual reproduction. Then there's meiosis, and then that produces the haploid spores. Okay, now ferns and other vascular plants, ferns and other non-seed land plant vascular plants are also dependent on water. Now, with ferns and vascular plants, we're switching from the haploid dominant stage to a diploid dominant stage. But yet the non-seed plants are dependent on water because the sperm essentially needs to swim to the egg, okay? And what we're going to be learning is that it was the seed that essentially freed plants from this dependence on water for reproduction. So let's start with a fern that we'll find in a forest growing in the understory of the forest. This is a mature sporophyte. This is a diploid individual. You have the sporangium on the underparts of the leaves. In this diploid phase, we then have meiosis, and you have a germinating spore that is now haploid. This is a germinating spore that is haploid because we've undergone meiosis. This mature gametophyte is really small. It's about a ½ centimeter wide, this heart-shaped structure. And on this individual, we have both the sperm and the egg cells. So the sperm are released and they swim, given this, there has to be water on this gametophyte. This has to be in a moist environment for that sperm to swim to the archegonium, where it finds the egg. This is where fertilization happens. You have a diploid embryo. That embryo grows up out of the gametophyte, it has a sporophyte here, and you then have the sporophyte that's diploid completing the life cycle, all right? So now with vascular plants, we're seeing the diploid phase is dominant, whereas in bryophytes, we were seeing the haploid phase is dominant. And what we're going to see is that with the seed plants, that all of the land plants leading up to seed plants, including some of the vascular plants, for example, ferns, are dependent on water for reproduction. All right, and normally this would be a think, pair, share moment. It would give you all a stretch and to talk about this. And so remembering with the Gametophyte, the haploid phase dominant, and the diploid phase being attenuated, shortened, then with the vascular plants, we see the diploid phase dominant, something we're more familiar with in terms of animals, where most of the life cycle is spent as a diploid. And a haploid gametophyte is small in body structure and also a shorter period of life cycle. So we could ask, what problem is this sporophyte dominant solution to? So what do you think are the advantages of having the sporophyte dominant solution? So I'll give you a moment to think about that. Okay. So there are a couple of things. So the first is that the diploid isn't nearly as sensitive to deleterious mutations. If you're haploid, you only have one copy of the gene. And if there's a deleterious mutation at that gene, there's no other copy to offset that deleterious mutation. So this can lead to trouble without having that other gene copy. Now, the sporophyte is free to get larger. It produces spores that are not dependent on water. Now, the gametophyte in the fertilization is dependent on water, but now the sporophyte can get much larger and be able to have more efficient capture of sunlight for photosynthesis. Now, the gametophyte is tied to water because the sperm needs to swim to the egg on the gametophyte. So competition for light is going to select for taller plants, and having taller plants is going to allow a more effective dispersal of spores. Okay, so thus far in thinking about the evolution of plants, we've been thinking about terrestrialization, the evolution of cuticles, stomata, a vascular system, leaves, a shift to a diploid dominant phase, and I listed leaves there twice. Sorry about that. So there should be no second leaf, even though leaves evolved twice in vascular plants. But now we're going to look at the seed. So everything in the phylogeny that's not basically this common ancestor and this clade are reproducing by spores. So all these plant lineages are reproducing by spores. Everything here are reproducing by seeds, okay? These are the seed plants. Now, the seed is great. It contains an embryo, nutritive tissue, and a protective coat. If this reminds you of anything, indeed, yes, it's just like the amniotic egg. And evolving seeds is crucial in the terrestrialization of vascular plants. It allows plants to colonize and reproduce in a much wider range of environments. It's because its reproduction is not tied to water. And also, seeds have dramatically increased ecological interactions between plants and animals. That is, seeds have are dependent on animals for fertilization as well as dispersal of seeds, and many animal groups are dependent on the products of seeds, that is, using seeds for food. So animals are providing critical seed dispersal and utilizing seeds as food. And then this will be extended to fruits, which are the product of flowering plants. Okay, now there are 5 major living lineages of seed plant lineages. So this is really not hard to learn. There's only 5 groups. We have the cycads with about 300 species. There's the cnidophytes with about 95 species. This is a really weird plant called Welwitschia that essentially is found only in the southwestern part of Africa. So areas like Angola, Namibia, for example. We have the conifers, which are all of your pines, for example, and sequoias. about 600 species. And then there's ginkgo. The ginkgo is only a single living species. There are a number of ginkgo trees on campus, including this lovely one in the TD courtyard. This picture was provided by Professor Liu, the head of TD College. And this they get this really incredible golden yellow color, the leaves in autumn. And then by far the most, by the way, there's only one living species of ginkgo. And then we have by far the most species-rich group of plants are the angiosperms, the flowering plants, with about 257,000 living species. So 5 major groups: cycads, ctenophytes, conifers, ginkgos or ginkgo, and flowering plants or angiosperms. Now, in the short lecture about the fossil record, I talked about how angiosperms were this really remarkable group that are very species rich today, but have this incredible appearance and dominance in the fossil record. And what we're looking at here, this would be the percentage of any taxonomic group at this period of geologic time. So if we're looking at in the Jurassic, conifers comprise about a quarter of all plant fossils. The cycads, which are about 300 species living today, plus conifers are about 50% of all the plant fossils. Other seed plants bring that total up to 75%. And then this other group here makes up the difference to 100%. But as we go through geologic time, as we get to the middle part of the Cretaceous, in the Cretaceous, we start seeing fossils of angiosperms or flowering plants. But in the middle Cretaceous, angiosperms, in a very short period of time, end up dominating the species diversity we find in the fossil record at these geologic times. And you could see these other groups, which were much more species rich in the fossil record, their diversity declines. And the origin of flowering plants is what Darwin called an abominable mystery, in that they're really distinctive plants. There's just a ton of derived features, and they appear quickly in the fossil record, and they essentially overtake the earth. And so here is a seed plant evolution through time, so to speak. So here we are, the geologic time scale. We could see the rise of seed plants in the latter part of the Paleozoic. Then through the middle part of this, the gymnosperms are really dominant. And then angiosperms attain their dominance towards the end part of the Cretaceous, really starting in the middle part of the Cretaceous here. And what we see is the fact that today, angiosperms are dominating species diversity, and we see that domination of the species diversity starting about 90 to 100 million years ago. So even though angiosperms are relatively late comers on the scene, they are clearly the group that has been hugely successful among the seed plants. Now, what really characterizes the angiosperm, of course, is the flower. So the flower not only provides is the place of the reproductive structures of the plant. Don't worry, we're not going to ask you to identify the parts of the flower. That is the male and female structures. But what's really important that I want you to take away is the carpel with regard to the flower. The carpel is the distinctive feature that encloses the seeds and forms the fruit. And this really is the innovation. So flowers are allowing for unique mechanisms of pollination and fertilization. That is, utilization of other organisms for pollination, as well as the ability to essentially wrap up the seed into a structure that becomes the fruit. Now, this not only gives physical protection to the seed, but by having a fruit, it could induce dispersal of the seed by animals that are going to eat the fruit, move to another spot and defecate out those seeds. or having fruits that allow for different modes of dispersal, such as wind dispersal, or fruits that actually stick to animals and will fall off at like a burr that will fall off at a place away from the parental plant. Now, we talked about how seeds were enabled a lot of interactions between animals and plants. Fruits really up that interaction to a large extent. So we have different kinds of fruits that are facilitating wind dispersal, fleshy fruits that are facilitating animal dispersal, and even explosive fruits where they get to a point where they dry, and then essentially they'll pop and will then just physically disperse those seeds away from the parent plant. Okay, now moving on, looking at the phylogeny of eukaryotes, let's take a look at fungi. So historically, botanists not only studied plants, but they also studied fungi. And in many previous old classifications, fungi and plants were always grouped together. But with the advent of phylogenetics, and particularly using DNA sequences and genomics to infer phylogeny, it's quite clear that fungi and animals share a common ancestor relative to all other eukaryotes. All right, now we'll talk a little bit about fungi. They're really important in human health. They're obviously important in disease control through the production of antibiotics. They're super critical in terms of ecosystems, that they are critical for decay and turnover of organic products. And they also are important food in terms of yeast for making of beer and bread, as well as mushrooms for food and medicinal, if not also recreational activities. Now, fungi, they have chitin in their walls. They store glycogen, and they can synthesize lysine, and they're heterotrophic by absorption, which essentially they digest their food around their body structures. So they digest the food outside of the body structure, and they're absorbing the nutrients after their actions of digestion. So basically a mycelium is the basic structure, is a structure of fungi, and the basic structure of mycelium is a hyphae. Now, fungi can be really big and old. In fact, one of the largest and oldest living organisms is a fungus. And I went ahead and highlighted here that the individual of this fungal species occupies 15 hectares and weighs in excess of 10,000 kilos. And it's remained genetically stable for about 1,500 years. So fungi could be really large and long-lived. Now, in terms of thinking about the diversity of fungi, what I really want to do here is I'm more interested in thinking about the unique aspects of how some fungi carry out sexual reproduction. And most of this lecture, most of the last part of this lecture on fungi is more just to give you an overview of fungal diversity. And as I said, in particular, we're going to be focusing on some of the not bizarre, but let's say unique strategies in terms of diploid and haploid, and this really interesting dikaryon stage that we see in the sac fungi, the Ascomycota, and the club fungi, the Basidiomycota. Most of us are most familiar with the Basidiomycetes. These are what are the mushrooms, and we're probably familiar with the Zygomycota because we see that if our bread gets moldy. Okay, now I just want to mention that there's a fungal group called chytrids. These are aquatic fungi, they're motile, and they have been implicated with what is a dramatic decline of amphibian abundance and diversity globally. And it's thought that these chytrid fungi, they're infecting the amphibians. They're a skin infection. And fungi or amphibians do a degree of their respiration through their skin, not just through air breathing through their lungs. And the chytrids are thought to interrupt the oxygen intake and are having a dramatically negative impact on amphibian abundance and diversity. Now, the Zygomycetes, these are your bread molds and other relatives. So typically, if you find some moldy fruit in the back of the refrigerator, these are Zygomycetes. They have no motile cells, okay? And the Ascomycetes are your cap fungi and relatives. So some, what we think of as mushrooms like morels, are actually Ascomycetes, and they're different from the Basidiomycetes, which make your mushrooms. And the Basidiomycetes are your mushrooms and puffballs. And just to show that there's quite a bit of diversity in the secondary compounds made by fungi. So while some fungi are really yummy and palatable, such as these morels, others are quite noxious and actually poisonous to humans. And then, of course, there are some mushrooms that, as I love this figure legend taken from this textbook, it says mushrooms that make you feel strange. So if you're going to mushroom hunt, you should definitely know what you're doing. All right, now what I want to do for thinking about reproduction in fungi, particularly the Basidiomycetes, and this applies to the Ascomycetes as well, is in the way that we were looking at the Bryophytes and the vascular plants, and what is going on with regard to the ploidy at the different life stages, all right? So one thing that's actually pretty cool to do is if you find a mushroom, you know, if you're walking around outside and you see the fruiting body of a basidiocarp, a mushroom, take the cap of the mushroom, and if you bring it home, you know, set it on a piece of paper or an index card and just put it on the windowsill, and let it set for a few days, and eventually the cap is going to dry off. And what you're going to find as you lift it up, you're going to have a nice impression of what will be what are called the gills of the mushroom cap. So essentially what's going to happen is the spores are going to be falling off of these gills, and you'll be able to see it on the index card. So what's going on with these gills? Well, these spores that are falling off on the edges of the gills, they're actually haploid, okay? So this is looking at the edge of the gill, and you can see these haploid spores are about to be released from the edge of the gill. Now, this is probably going to be the most unusual diploid sexual reproduction that you're going to encounter in studying the history or the biodiversity of eukaryotes. Okay, so blue is haploid, orange is diploid, and green is dikaryotic. What is dikaryotic? Well, dikaryotic is that you have two haploid genomes in a single cell. Now, these genomes are not interacting with each other, but they are basically dikaryon. It's not a diploid. It's two haploid genomes enclosed in a single cell. So what ends up happening is you have basically the gills are lined with these spores, and basically on the gills, these are dikaryons. So the cells in the gills have two haploid genomes. There is then fertilization where the fused nuclei, they fuse, so they become a zygote. So they're diploid for a very short period of time. So here they're dikaryotic, two haploid. Then they fuse, now they're diploid. Then they undergo meiosis, all on the gill of the basidium, the mushroom. Then you have these basidiospores that are haploid. Each cell has a haploid genome. These spores are released. Then the spores germinate to these mycelial hyphae. Remember, hyphae is the primary structure of a fungus. And you have different mating types that are compatible with each other. Yes, within the species. Then these mating types will fuse together and they will make a dikaryotic mycelium. So the mycelium here is dikaryotic. Remember, cell that has two haploid genomes. Then this dikaryotic mycelium will then form the basidio fruiting structure, the mushroom. And then the life cycle is then completed. So mushrooms and ascomycetes, the cup fungi, are diploid for a very short period of their life cycle. They're haploid for part of their life cycle, but much of their life cycle, they are a dikaryon, where the cells contain two haploid genomes. Now, many fungi are parasites of insects. And we talked about the cordyceps in the first lecture, and I had the link to that bizarre zombie ant video. And some species of fungi you could think of as being hunters, in that some fungi have these lassos that are designed to capture, digest, and absorb the nutrients from soil-dwelling nematodes. And many insects eat fungi, and many have evolved symbiotic mutualistic relationships with fungi. For example, fungus farming and leaf-cutting ants, where ants will cut up leaves, take them to their colonies, inoculate them with a fungus that grows on the leaves, and then they are then eating on the fungi that grows on the leaves. Now, lichen it's not a fungus, but it's a symbiotic relationship that involves a fungus, usually an ascomycete, and an algae, a green algae, or a cyanobacterium. So a lichen is a composition of at least two different organisms, that one being a fungus and the other being some type of photosynthetic organism, whether it be an algae or a cyanobacterium. Now, there's a lot of symbiosis between fungi and plants, and this is through mycorrhizae, which are structures of the fungus that either are associated with the outside of the plant, and actually some plant roots, this symbiosis is very intimate, to where they actually have compartments in the root that facilitate growth of the fungi in the root, and these are called endomycorrhizae. And the fungus provides a lot of benefit to the plant in terms of helping the plant deal with nutrients that the fungus is making through it or through its external absorption. And then basically it's helping the plant obtain water and nutrients, or both are benefiting through the obtaining of water and nutrients. And it's thought that the connection in the high species diversity of angiosperms of flowering plants we see, in part, is been facilitated by the diversification of ecto and mycorrhizal fungi, whereas there's symbiotic relationships between the fungi and plants. Okay, so in lecture 8, we talked about the phylogenetic diversity of plants. You should be able to articulate green plants, land plants, vascular plants, seed plants, and then the 5 major groups of seed plants. You should be able to articulate the origin of green plants and think about algal diversity, that is a paraphyletic group. Think about the challenges plants face in the colonization of terrestrial habitats. Think about different life cycles of plants, the diploid and haploid dominant phases in particular with the non-vascular, vascular plants, and the evolution of the seed. Think about the traits that may have facilitated angiosperm's dominating plant biodiversity that we see today, as well as see that change in the fossil record. And think about fungi, their structures, the different aspects of their life histories and life cycles, and their importance to biodiversity. So Bio 104 students, until next time, be safe and be well.